Hi, everyone. First of all, I would like to play a game with you. So please raise your hand if you have a good time here. That's the first question. Wow, that's really good to see. Cool. Okay, next question. Who would like to be a physician, a doctor who treats patients? Oh, many of you. Very nice. Okay, let's move on now to a more complicated one. Take a moment and think about the virus. Okay. Now, raise your hand if you thought of a virus that caused disease. Yeah, many of you, right? That's pretty normal because we hear a lot about these viruses. But yet, not all viruses like this. Actually, there are some viruses that can cure disease instead of causing them. So these viruses are called bacteriophages, or phages for short. So the special feature of these viruses that they eat bacteria for breakfast. Okay, so some of them are so special that they can kill superbugs. Have you heard of superbugs before? Yeah, the superbugs are those bacteria that become resistant to antibiotics. Some of them are so dangerous that they become resistant to all antibiotics that physicians can use in the clinic. So that's a serious problem, because then these infections become permanent. It's not possible to cure them. So first of all, yeah, if you may become a physician, there is a very high chance that you are going to face with this problem in the future, primarily because these resistant bacteria spread in hospitals. Therefore, those patients who frequently get hospitalized has a very high chance to acquire such infections. In fact, 1 in 30 patients in general acquire hospital-acquired infections, meaning that they become more sick in the hospital. So that's a serious problem. Actually, it's so bad that the prediction is that by 2050, resistant bacteria may become the leading cause of death if action is not taken. So who are susceptible? Those who are immunocompromised, let it be from a genetic disease, or who become immunocompromised due to therapy, like chemotherapy, or maybe from a viral infection which require intensive care, like COVID infections. So if these infections don't respond to any antibiotics, then there's the application where phages may become an option in the future. Okay, so now there are hundreds of case studies all over the world where patients receive a phage-based therapy from physicians. Most recently, we managed to be involved in such case studies, and the first patient who received a phage from Szeged is a Portuguese girl who has cystic fibrosis. She received this treatment 4 weeks ago, and now she is much better. So I'm going to show how does it work through her example. So cystic fibrosis is a hereditary disease. Out of 25 people, 1 carries a gene, a CFTR gene, that contain a mutation that makes this gene dysfunctional. Okay, so meaning that in this auditorium, there may be about 10 people who carries this CFTR gene variant. It's not a problem as long as this carrier doesn't want to mate and have a child with another carrier, because then there is a 25% chance that the child will have cystic fibrosis. And this is a serious disease. The life expectancy of a cystic fibrosis patient is about 30 years in the EU, maybe about 20 years in Hungary. This is primarily because they are very susceptible to lung infections. They have very thick mucus layer within their lung, which makes them very difficult to breathe. And these bacteria, like Pseudomonas aeruginosa, likes to live in this hostile environment. So the problem is Pseudomonas aeruginosa is one of the superbugs. And it can happen that the patient acquired the infection in the hospital, unfortunately. So this happened with this Portuguese girl as well. And fortunately, her physician heard about phage therapy, so he put a phage alert to the web that we saw, and we reacted that we may have a phage to treat this patient. So the sample, the bacterial sample isolated from this child, was sent to Szeged, where in a special laboratory, my colleague isolated a phage that worked against, that killed these bacteria. Then we sent this phage to Belgium, to another special laboratory in a military hospital, where experts were made a preparation, pharmaceutical grade, meaning that they made a phage preparation to a medicine. Okay, then they sent this phage back to Portugal, where this child received the treatment. We were very happy about this because this was our first case study, and fortunately, girl is better, but this whole process took about 6 months. Okay, so 6 months is a huge time when you have an antibiotic-resistant infection. Most of the patients actually die in this time period. So our aim was to reduce, or our aim is still to reduce this time, the 6 months, to 1 day. How is it possible? So the whole process can be reduced to 1 day if all of these, the production, delivery, and so on, happens before the infection established, right? Because there is a huge problem of phage therapy. The phages have very narrow host specificity, and this entire process needs to be repeated with every different bacteria. And unfortunately, there are hundreds of superbugs, hundreds of different types of superbugs that require a different bacteriophage. That's why it is very important to decrease this time to a minimum what is possible. Okay, so how can we decrease the time to 1 day? That's the question. So if all this process happens before the infection, right? But for that, we need to be able to tell which bacteria variant will infect a specific patient in a specific hospital in advance. So the major question was, what is the geographic distribution of superbugs to inform phage therapy? For this question, we used another bacteria called Acinetobacter baumannii. This is another deadly superbug. It kills about 5,000 to 10,000 people annually in Eastern Europe. So what we did, we took 10,000 samples, 10,000 Acinetobacter baumannii genomes, so all the DNA that is present in this bacteria, and used the sequence to build a so-called phylogenetic tree. This is basically a family tree. It tells who is a close relative of who. And for the Eastern European variant, and that tells us what type of variants of that bacteria is present in a certain geographic region. And then, against the variants that spread in Eastern Europe, we find phages that can target these bacteria. Mostly we isolated these phages from wastewater, from communal wastewater treatment plants, because this is where all the bacteria goes from cities, right? And, and then we integrated all this data into a model that tells us that in which country, city, and hospital, which type of this Acinetobacter baumannii infection is present, and which bacteriophage shall we use to treat those infections that are present in these hospitals. So to show you a little bit more data on this plot, you can see that in Eastern Europe, which is the green line, 90% of the infections is caused by altogether 11 types of Acinetobacter baumannii infections. Okay, so Acinetobacter baumannii strains. So this 11 type causes this 5,000 to 10,000 deaths annually in this region. Moreover, we find that using statistical analysis that within a country, it is 20 times more likely that an Acinetobacter variant causes the next infection that is already present in that country compared to those that are outside of that country in a 6-year time window. Meaning that in 6 years, we can tell which variants are causing 95% of the infections. Unfortunately, this pattern then disappears on a longer time scale, like more than 6 years, because of international transmissions. Moreover, we see a similar pattern on the level of cities. So if it is 4 times more likely that an Acinetobacter variant that is present in a certain city is going to cause infections in a 2-year time window as compared to those variants that are not present in that city, but present in the country. In Hungary, there are 6 types of Acinetobacter baumannii. So we can predict quite nicely that which are the variants that in specific cities are going to cause these infections. And we can prepare phages in advance. Okay, so in this heat map, we can show those 15 phages that we isolated against the Eastern European Acinetobacter baumannii variants. Those are the rows, those are 15 phages. And here you can see the 11 variants that are responsible for 90% of the infections. The darker the blue, the more efficient is the phage to eliminate the bacteria in vitro. Okay, so as you can see, 100 isolates on this heat map, about 95% of those isolates we can now target with at least one phage. Of course, we don't want to stop here. We would like to take these phages to the bedside of the patients. Finally, I'd like to show the laboratory where we work. So in the last 2 years, we built a completely brand new laboratory with very high biological safety level. It is important considering that we work with pathogenic bacteria. And as you can see, the team is quite young. There are PhD students in the laboratory and also medical students like Dorina, who presented in the previous section. So the take-home message is that if you take your studies seriously, in a couple of years' time, you can be involved in research that has an impact. Thanks a lot for your attention.
Thanks a lot, Bálint, for this very interesting presentation.